Hello. Oh, no. Okay, hello everybody. How are you? Can you see me? Can you hear me?
Yeah, we can.
Okay, excellent. My hands are so cold that I'm having trouble moving the cursor. So, yes, you don't see the lecture slides?
It's only the notes that are on Brightspace, not the slides.
Yeah, the slides should be on Brightspace. They're
No,
not there?
only the notes. No slides, only notes are there.
Oh, no. Oh, I thought I had checked. Let me see again. Oh, tell me. Can you see the slides now?
Yes, they're there now.
Okay, great. Thank you. Okay, to chat up. Okay. Okay, fantastic. I just forgot to no, what doesn't this work? There. Okay, let's start class. It's very cold where I am, and my hands are sticky or not sticky enough. Okay, so today what are we going to do? We're going to talk about the cells of the nervous system. So here we have the cells. I just want to be able to do this. There. And then I will go down here. So you see here two different types of cells. Actually, just the names. The neurons, which are the most important cells in the CNS, or the nervous system, I should say. So you see the neurons with the cell body and the projections, some are dendrites, some are axons. But then there's other cell types that are there to maintain, to help maintain the neurons. And those cells are known as glia cells. And there's several types. One of them is shown here in green, and it's an astrocyte. And of course, it's not green. It's shown in green because it's stained with a specific dye just for astrocytes. Okay, so let's go to the neurons. Okay, here you see a neuron. And you can see right away that it's a very, very complex cell. And no wonder that it lost its mitotic abilities. So you see the cell body up here with the nucleus. They have one nucleus. And you see that it's highly branched. And these are what we call the dendrites. And they are branched like a tree because they are there to make connections with other neurons. So you know that there's these networks of neurons are very, very complex. And you go from one neuron to another, to another, to another. And one neuron can communicate with more than another neuron, more than one neuron. So the networks are very complex. So these short branches that we call the dendrites, they bring the signal from the outside towards the cell body. So what I'm talking about is the electrical signal, the action potential. Remember, they make synapses with other neurons, and then the action potential is transmitted from the presynaptic neuron to the dendrite of the postsynaptic neuron. So they have branched, and another property is that they are not myelinated, and usually they're short. And then you see here a main projection, which is part of the cell. So this is one cell, one continuous cell, and you see that the axon is very long. And once they let who can see our messages recording on? the recording stopped? I don't think so, because I don't have control over it. Wait, maybe it might be me. Okay, maybe someone else please confirm that the recording is on. Okay, so then you have the axon, which is quite great. The axon is very long. And remember, there has to be a lot of movement all the way from the cell body all the way to the axon terminal. And see, here you see another neuron, and you see it's shown in yellow. You see the axon, just partial axon. You see the dendrites, and they are synapsing with the axon terminal of this blue axon. So there's a lot of interaction and movement going from the cell body all the way to the axon terminal, and from the axon terminal all the way up. So it moves in both directions. So remember, muscle cells and neurons are the only ones that are excitable. That means that they can generate an electrical signal, an action potential, once they're stimulated, but the stimulus has to be strong enough. So neurons, when they die, they're not replaced. There's no regeneration in most cases. And the axons can be very, very long, like going from the waist all the way to the tibial, like the sciatic nerve is the longest one. Or they can be short also, especially the interneurons. We will talk today. You will know very well, you will have a very good idea what is an interneuron. And the impulses, the electrical current that goes along, that travels along the axon, can go very, very fast, sometimes up to 218 miles per second. Okay.
Huh?
Yes? Question? Okay. So
is it per second or per hour?
per hour, per hour. Second would be impossible, right? It's per hour. I don't know why this thing here. Okay, well, it's per hour. I'm so sorry. Okay, so what is a synapse? That's the area where the presynaptic neuron will meet the postsynaptic neuron, like a neuromuscular junction, except of being a junction between a neuron and a muscle fiber. You have now a junction between two neurons. But never forget that they do not touch. They are not in physical contact. There's always a space that's the synaptic cleft. And we need a lot of different factors to maintain the neurons in good condition, and these are known as trophic factors. Since the dendrites are not myelinated, will it carry the action potential slower than the myelinated axon? Absolutely. But now remember that the dendrites are short, right? So, but absolutely, the speed of the action potential in unmyelinated dendrites, and there's also some axons that are unmyelinated, and the speed of propagation under these conditions is much, much slower. Okay. So neurons have different shapes. Here's a neuron that's known as the pyramidal cell. It can be in the cerebral cortex. And you see that it's simpler. It only has much fewer dendrites and the axon and the synaptic terminals. So not all neurons are the same. The design is very different. The neurons that have most dendrites are the Purkinje cells. And you will see an image of a Purkinje cell. These Purkinje cells have nothing to do with the Purkinje fibers in the heart. So these are neurons, Purkinje neurons. And where are they located? They're located in the cerebellum. The cerebellum is a small part of the brain, and it's evolved in precision. So let's say a basketball player has a very well-defined and developed cerebellum. So those are the Purkinje cells. Okay, now let's look at neurons. So here is a beautiful neuron. Well, neurons are much more beautiful than what is represented here. But you see the cell body, and you see the axon, and you see the dendrites. And you see here these red arrows that indicate that the electrical current is going to be conducted from the end of the dendrite towards the cell body. So this would be the postsynaptic membrane of the dendrite towards the cell body. In the cell body, you have the usual components of cells, nucleus, one nucleus. They have many mitochondria. They also have Golgi and lysosomes. Remember, lysosomes are the organelles that are supposed to digest macromolecules going from carbohydrates, lipids, and so on. But there are other components that are specific to the neurons that are located in the cell body. These are Nissl bodies. What are Nissl bodies? So Nissl bodies are shown here. They're almost like rough ER. So they're areas within the cell body of the neuron where there's lots of ribosomes and where there's lots of protein synthesis going on. So here, these aggregates or formations of these ribosomes with the ER are known as Nissl bodies. Then the neurons, besides having tubulin and actin, remember the cytoskeleton, they have other components of the cytoskeleton that are known as neurofibrils. And these additional cytoskeleton components are very important because of the movement that goes back and forth from the cell body towards the axon terminal. They also sometimes have dark spots that are known as lipofucsin. What is lipofucsin? Lipofucsin is a remnant of products that are digested by lysosomes, but that are not completely digested, and they stay. It's like, aging spots, dark spots that people have on their skin. They also contain lipofucsin, or they contain this kind of what happens when the material is digested and not completely. Okay. Questions here. Do you know how they measure the speed of the neuron? Ooh, that's a very good question. So it has to be with electrodes, right? So you put electrodes on one place and electrode in another place, and then they stimulate the neuron, and then they have very precise computers and apparatus to measure this. But you have to have two electrodes, one where the stimulus is added and the other one in an area of the axon, and you see how the electrical signal, how long it takes to go from one place to the other. Okay, so cell body. Now we did the dendrites, we discussed the cell body. Now let's go to the axon. So the axon, you can see here, it's very long. You see the axon terminal that ends in these, what we call synaptic knobs. And usually one neuron only has one axon, but sometimes they can bifurcate. They form what we call collaterals. So that means instead of just having one axon all the way, you have another one that can come out, and that would be a collateral. Okay, so what is important in the axon? This area here is very important. This is where the cell body meets the axon. And this is known as the axon hillock. So hillock is a hill. It's a little bit elevated. And why is it elevated? It has a high concentration of voltage-gated sodium channels. So keep this in mind. Voltage-gated sodium channels. And it's also known here, the trigger zone. So neurofibrils are part of the cytoskeleton, additional cytoskeleton present in neurons, such as neurofilaments and so on, and vimentin. So here, this area is very important. It's known as the axon hillock. And right here is the trigger zone. So what happens? What is the importance here? So you have the dendrites feeding electrical signals to the cell body. But these electrical signals of the dendrites are not so strong. So the resting membrane potential of a neuron is around minus 70 millivolts. That's when the neuron is quiet, is down, is not being activated. Remember, in the muscle, it was minus 90. Here we have it at minus 70 millivolts. Okay, so then the electrical signal comes from the dendrite, and it's usually weak. So it might bring the potential from minus 70 to minus 60, from minus 70 to minus 65, from minus 70 to minus 60, and if it doesn't change, nothing happens. That electrical signal will die out. So the change in potential has to reach a threshold. That threshold in a neuron is minus 55 millivolts. So until the potential in the cell body is not changed in a way that it reaches minus 55 millivolts, the action potential will not be triggered. So what happens is each dendrite can contribute to a little bit of change in potential, which is going to be added up. And once it reaches this area where there is highly enriched in voltage-gated sodium channels, so there the sodium channels open, and then the action potential is an all or none response. Then it goes down the axon. That's right. It's the trigger zone is only activated when it reaches a certain level. Let me just see something here. So that's right. So until it reaches, remember I told you, so the beauty of this design is that we do not respond to all changes in our environment. So the changes have to be strong enough to induce, to trigger, to change the voltage in the neuron from minus 70 to minus 55. Until it reaches minus 55, it will not be converted to an action potential and travel down the axon. So minus 55 is the threshold that is required for the action potential, the resting membrane potential to change, for the action potential to be travel down the axon. Okay. So the sodium channel must be open for the axon to work. It's not just one channel, it's many channels. So all those voltage-gated sodium channels will open up, and then the voltage goes up and it goes down the axon. It's an all or none response. And remember, it goes from minus 55. That is the triggering signal. But then it has to go to plus 30. So that's why you have so many sodium channels here, Once that threshold, the minus 55, is reached, then all the sodium channels that are here, the voltage-gated sodium channels open, increase the potential to plus 30 millivolts, and then the action potential travels down here. So you go from minus 70 to minus 55 threshold, action potential triggered, reaches plus 30, and then it will go down here, down the axon. What is the first, the axon, helicus, or the trigger zone? So this is the area, the axon hillock is an area here where the cell body meets the axon, where there is a high concentration of voltage-gated sodium channels. Okay, so what is RER, rough endoplasmic reticulum? So within the axon, there's protein synthesis going on. So we have ribosomes that are here and that produce proteins right in the axon. Okay, so the question, so how does the plus 30 go back to minus 70? Remember, in the muscle, what you need potassium. Potassium channels open up, potassium leaves, and then you get repolarization. So you get depolarization going from minus 55 to plus 30, and then that corresponds to the opening of voltage-gated sodium channels, and then it goes from plus 30 to back to minus 70 with the opening of potassium channels. Right, so the potassium channels are along the axon, not so much concentrated here, because this is where the trigger zone is located. Okay, so in the axon, you also have cytoplasm that's known as axoplasm. The membrane is known as axonema. And then you have the axon collaterals that allow this neuron to communicate with more than one postsynaptic neuron. And then you have the axon terminals. So here, what is represented is synaptic knobs. But remember that with smooth muscle, the axon terminal ends up in varicosities. Okay. So let's move on. So here you see the trigger zone. This is very, very important. Okay, and here, so the question is, so cardiac and skeletal have the knobs and smooth have varicosities. Yes. So here you see another drawing of a neuron with the dendrites, and you see the Nissl bodies, these dark areas that contain ribosomes. And then you see here the trigger zone. Here's the axon. You see a collateral. And now here you see myelin. So here what is interesting is that each of these areas that are covered with the blue, this is a cell. It's a myelinating cell. It's known as a Schwann cell. You see here, this is a Schwann cell that is enveloping the axon of the neuron. And you see here the nucleus of the cell. And what happens is that this cell, first it approaches the axon and then envelops the axon and starts to go around and around and around the axon, pushing out all of the components, the other components of the organelles of the cell go out to the outer layer, including the nucleus. So you have sometimes hundreds of these layers formed by one cell, by one Schwann cell. And this is how the axon is insulated. But you see that it's not a continuous myelin sheath. It's just one, goes 1 to 2 millimeters along the axon. So collaterals are just an axon that bifurcates and forms here a bifurcation so it can reach another neuron. Okay, so here you see now in red means the action potential, or the change in electrical potential. So you see the incoming signal, that's very important. It's coming through the dendrites. Now remember that this is going to be more complex because some of these might be excitatory, others could be inhibitory. So that means that you have a plethora of signals converging on the cell body of a neuron. Some are releasing neurotransmitters that are excitatory. Others are releasing neurotransmitters that are inhibitory. Remember, the inhibitories put a brake. And it's adding up. All of those signals is what determines at the trigger zone whether the action potential is going to go down the axon. So the change in potential here has to reach the threshold minus 55 millivolts so that it can go down the axon. So what happens here in the cell body is a result of the signals, the electrical signals that are conveyed by these dendrites, some being excitatory, some being inhibitory, and the sum of all of these has to cause a change in potential from minus 70 to minus 55 for the action potential to go down the axon. Okay, so now here you see synaptic knob. So what's happening is pretty similar as what happens at the neuromuscular junction. Now, instead of having a motor neuron and a muscle fiber, now you have two neurons. So you have the axon of the presynaptic neuron, here shown in pink, and here would be the dendrite of the postsynaptic neuron, represented in gray. So you see the usual, the familiar components, and specifically the synaptic vesicles that contain the neurotransmitters. But now at the neuromuscular junction, the only neurotransmitter that is located in those synaptic vesicles is acetylcholine. Here, when in these synapses between two neurons, the synaptic vesicles can contain a plethora of chemicals that you will learn soon. Some are excitatory, some are inhibitory. Okay, and you see the synaptic cleft and you see the postsynaptic membrane. So this you are pretty familiar with because in comparison to a neuromuscular junction. And now here you see that instead of having a synaptic knob, now you have, for the smooth muscle, remember, we have the axon terminals end on varicosities. So the varicosities are these like bead-like structures that are represented here at a higher magnification, and the green represent the synaptic vesicles. So this allows for a shower of neurotransmitters to be released all at the same time over the top layer of this smooth muscle layer. Okay, so let's move on. Now, let's learn a few terms here. So what is a nerve and what is a tract? Okay, so when we talk about nerves and tracts, we are not talking about the cell body of the neuron. We are talking about the processes, the axons, right? So they are kept in bundles, just like the muscle fibers are also kept in bundles, right? So here, and remember the perineuron is in circles, so each of these small circles represents one axon. And you see that they are arranged in groups surrounded by a perineurium. So these, when they are in located in the central nervous system, they're known as tracts. When they are located in the peripheral nervous system, we call them nerves. So that's the difference between a nerve and a tract. So here you see the epineurium that envelops either a nerve or a tract. So you have the same layers, those three same layers of connective tissue, either around a nerve or a tract. Then you have the perineurium around a bundle of axons, and then around each axon you have the endoneurium. Now here we have the ganglion, or ganglia for plural. So remember, this is the sensory neuron that has a cell body outside the CNS. And I was looking up what is the advantage of having the cell body outside the CNS of the sensory neurons. So one advantage is that they are very well separated. The sensory neuron and the motor neuron are very well separated. If they would all be enclosed in the motor neuron, it would be kind of confusing. They are also differentiated from different components of the nervous system. So ganglia, we're referring to cell bodies. Nerves and tracts, we are referring to the axons. Okay, so I hope that this is clear. So ganglion contains many cell bodies of sensory neurons. Okay, so we can classify neurons. There are different types. Remember, there are different types of muscle fibers, so there are different types of neurons as well. We can classify them structurally and functionally. So structurally means shape. This type of neuron is known as anaxonic because it doesn't have a preponderant axon. They all look like dendrites, but some of them will serve as axon. And these are present in the brain. Then you have this type of neuron that is pseudo-unipolar. Pseudo means fake or mimicking. So it's saying that this neuron only has one process, but it really has two. It has a dendrite and has an axon, but they are almost continuous because the cell body is pushed to the side. So these are the sensory neurons. So sensory neurons are pseudo-unipolar because it looks like they only have one process, but they do have a dendrite and they do have an axon. It's separated from the cell body, but the cell body is pushed to the side. And here you see the myelin sheaths, the Schwann cells. Then you have bipolar neurons. Here you can see that there's a big difference here. They have two processes, but here they look like they're contiguous. But here they're separated by the cell body. These are bipolar neurons that are located in the retina. Why isn't the myelin sheath one continuous casing? Why are there gaps in between? Well, can you imagine having to synthesize a sheet all along a neuron? That would be really, think about the sciatic nerve, all the way from the waist all the way down to the tippy toe. Also, another big advantage is, let's say you damage a certain area of an axon. So then it would be very hard to repair if the whole myelin sheath was continuous. Like this, if you damage just one part, then this Schwann cell can work with or the other ones that are in close proximity to repair. So several advantages. Okay, now we have the multipolar one. These are the ones that are most abundant. And you see the cell body with the dendrites, one axon, and you see the myelin sheaths. Or Schwann's cells only on pseudo-unipolar, no. Schwann cells, you will see, We are going to discuss that soon. Schwann cells are present in the peripheral nervous system. In the central nervous system, the cells that myelinate have a different name. You will see. Surprise. Okay, so how about the functional classification of the neurons? So we have three: sensory, motor, and association or interneuron. So these are the three major groups of neurons. Sensory neurons are the afferent. We talked about this all the time. They go from the receptors to the central nervous system. They have the cell body pushed aside. And we have sensory somatic, that they are involved in the conducting the action potential from receptors that are at the periphery, like the eye, the nose, and so on. And they're also involved in position. And then there's sensory receptors that are inside the body. So these neurons will be sensorovisceral, and the receptors are interoreceptors inside the body. So you have exteroreceptors, proprioreceptors, and interoreceptors. Then the motor neurons are those that carry the efferent message, going from the central nervous system out to the effectors. And what are the effectors? They're skeletal muscle, that would be the motor somatic neurons, or smooth muscle, cardiac muscle, and glands. And those would be the visceral. These would be autonomic. These are voluntary, the somatic are voluntary. The visceral are autonomic. And then you have the association neurons or interneurons. So these are neurons that are located entirely within the central nervous system. What do I mean entirely? I mean that the cell body, the axon, the dendrites, they're all contained within the central nervous system. So you know the sensory part of it is in the peripheral nervous system, part of it is in the central nervous system. The same for the motor, but for the interneurons, they're totally located within the CNS. And look here, there's 20 billion of them. They communicate, they transmit the electrical signal from one neuron to another within the CNS. Okay, so here I'm showing some examples of these interneurons. Here are the Purkinje cells, those that are present in the cerebellum and that are so important for acuity, for precision movement. So you see, look at the dendritic span or tree that is so abundant, and it is bringing signals into the cell body. So it's highly regulated and receiving a lot of different signals. So this is the Purkinje cell. This is another cell, a pyramidal cell. It's also interneuron, and it's present in the cerebral cortex, and this is involved in memory. So I'm not sure. So who are they, involved sensory neurons or just motor? You mean involve, you mean Schwann cells or interneurons? So interneurons establish communication among all different types of neurons, not specific to sensory or motor, different types. Okay? Okay, so here's the organization of the nervous system. So you have the receptors, sensory afferent, the cell body, then going in the gray matter of the spinal cord. Remember this cross-section of the spinal cord? The gray matter contains the cell bodies, the white matter contains the myelinated processes. Here you have the interneuron, and now here the communication is between a sensory neuron and the motor neuron. Here is the interneuron. So then the electrical signal goes out to the effectors. Okay, now let's look at the axon per se, which is a very complex kind of environment. because you have a lot of movement from cell body to axon and back. So what provides for this movement? So there are proteins that we call motors. Motors, they're like the cars. They're the motors that carry cargo either from the cell body to the axon terminal or from the axon terminal back to the cell body. So these proteins are known as motors. If they're motors, you can imagine they require a lot of ATP. So these proteins, one is kinesin. Kinesin is a motor protein that carries cargo from the cell body all the way to the axon. So this protein is a specific motor to carry cargo from the cell body all the way to the axon terminal. The other protein that goes in the opposite direction is dynin. Dynin carries cargo from the axon terminal all the way to the cell body. So you have these two proteins that carry cargo in opposite direction. So the enterograde transport can be slow or fast. Slow, it goes from 1 to 3 millimeters per day. Fast, in terms of cell transport, 20 to 400 millimeters per day. So this is the cargo for mitochondria. So you need mitochondria to come from the cell body all the way to the axon terminal, because you need ATP here also. And then the ones that go slower are enzymes, cytoskeletal components, and so on. Now the retrograde is always fast, 200 millimeters per day. And what goes towards this side? From this direction, I should say, from the axon terminal to the cell body. So products that come into the neuron at the axon terminal by endocytosis. Remember, endocytosis, things that come into the cell. The membrane invaginates and brings in products that are in the extracellular environment. So this is what is carried by dynin. And what are some of those? So, there's a lot of products that come in, nutrients, amyloid, beta, things that are good and things that are bad. So viruses, they're very smart. They come in through the axon terminal into the cell through endocytosis, and then they reach, because here there's no nucleus. So it will be very hard for the viruses to divide. So they need to be transported all the way to the cell body, where they take over the machinery to make more viral particles. So viruses such as herpes and rabies and bacteria such as tetanus, they come in through the axon terminals via endocytosis, and they are carried by dynin all the way to the cell body. Well, we can also use this strategy for gene therapy to treat the nervous system. So you could infect the neurons, or let's not say infect, administer viruses that are genetically engineered to have a specific gene that is necessary. So they would be, those genetically engineered viruses would come in through the axon terminal and be conducted via dynin all the way to the cell body, where then they could replicate and make the protein that is not mutated. Would calcium be one of them? I'm not sure what you mean, calcium. Calcium is an ion. It will not be transported by kinesin and dynin. Calcium diffuses, right? It doesn't need to be transported. I don't know if that's what you're referring to. Okay, so here you see again the dynin bringing a hook. So once the viruses or bacterium reach the cell body, they can spread to the entire, well, the cell body. What you mean, entire body of the humans? Well, the viruses in the cell body of the neuron, and they can go to other cells also. They're not not particular to the neurons. So they use the cell body of the neuron to make new viruses that then have to be released by exocytosis, and then through the blood they are spread to the rest of the body. But they need first the virus the viral particles that come in need to go to a cell body. It doesn't matter if it's a neuron or any other cell, so that they can make more of their own proteins and to assemble. And then they got released, and then they travel through the blood to other parts of the body. In retrograde, is all cargo taken in through endocytosis? Yes. But, there's some cargo that is here, like, let's say, for example, that's a good question. Let's say mitochondria that are here at the axon terminal, right? They're already there. They didn't come in through endocytosis. So organelles that are damaged and old at the axon terminal, they have to be carried by retrograde transport to the cell body, because that's where the lysosomes are. There are no lysosomes here, or if there are, they're very, very few. So most of the damaged organelles that are present at the axon terminal, they have to be carried to the cell body to be destroyed by the lysosomes. Why would the viruses enter to the axon terminal? Because endocytosis is more efficient at the axon terminal. And, it depends on the receptors. Remember that the viruses, they hijack our own proteins. So it depends on where those receptors that are hijacked by the viruses so that they can enter the cells. So that depends on the receptors. So we finish on time. Well, we still have 15 minutes. Thank you, Sophia. Okay, so here we go. So this is what I wanted to show here. And now, so here I just want to see that the highways within the axon are not only you have the microtubules, which are the ones that are thickest, cytoskeleton components, but you have other filaments like neurofilaments that help in this transport. Okay, I'm going to pass this. And this is just showing, this is the rabies virus, very, very complex. So when someone is bit, then the virus enters through the axon terminal and is transported via dynein all the way to the cell body. So you see saliva of infected animals, bites the skin, then at the synaptic knobs in the peripheral nervous system, the viral particles are transported to the cell body, which is in the CNS, and then they replicate, they assembled, and then they are transported down to the salivary glands, where they then can infect other individuals. Why is it that rabies is incurable? Well, it's treatable, but we still don't have, all these things. You think it's very easy. It's not. It's very, very hard to make vaccines. It's very hard to treat different diseases. It all depends on the success of the research. Okay, so here is the gene therapy. So the axon terminals are always part of the PNS. No, I don't know where you got this idea. No. Some of them, like for example, if you think about the interneurons, right? The axons of the interneurons are within the central nervous system. And you will learn, no, doesn't mean the axon terminals are always in the PNS. Not always. The axon terminal of the sensory neurons are in the CNS. Well, the slide said synaptic knobs. Where? Oh, let me see. Oh, I see what you mean. With the virus, right? Here. Here. I don't know, but right, the rabies one. Because it's at the skin, right? That's where the person is bitten at the skin. It's not, the person is not bitten in the CNS, it's bitten at the skin, and that's part of the axons there are peripheral or within the peripheral nervous system because they are at the skin, they're not at the CNS. But that doesn't mean that all axons are there or in this or in the peripheral. And this is showing the gene therapy, how you can also deliver viruses that are genetically engineered to deliver a gene that is not mutated. Okay, now let's move on to the glia cells. Okay, so neuroglia, there's different types of neuroglia. So now here we're talking about cells that are there to nurture the neurons, to help the neurons survive and be under optimal conditions. So, but there are different types of glia cells, you will see. So they support, nurture, and protect the neurons. They attach neurons to blood vessels, like the astrocytes. They provide a bridge neurons to be close to blood vessels. They produce myelin, some of them. They go through they can provide for phagocytosis to get rid of bacteria and so on that reach the CNS. You will see which kind of cells. So these are different types of cells that have different functions. And they're smaller than neurons. Neurons usually don't develop cancer because they do not have the capability of undergoing mitosis. They are in the G0 phase, but the neuroglia can, unfortunately, differentiate or develop into cancer. For example, here's an astrocytoma. That means it's an astrocyte that is that formed this cancer. Okay, so here we have the neuroglia cells in the central nervous system. So there's more neuroglia cells in the central nervous system than in the peripheral nervous system. In the peripheral nervous system, we only have 2 types. Here we have 4 types. Okay, let's start. Well, here are our beautiful neurons. Right here, these are the cells that myelinate neurons in the CNS, in the central nervous system. They're known as oligodendrocytes. And you see that their shape is very, very peculiar. So they have these processes, and one oligodendrocyte can form these myelin sheets along the axon. So if you look at the myelination in the peripheral nervous system, where you have the Schwann cells, you have one Schwann cell, another Schwann cell. So one Schwann cell is dedicated to a section of the axon at the peripheral nervous system. Here in the central nervous system, you have oligodendrocytes instead of Schwann cells. And these oligodendrocytes, you see that the nucleus is far away from the axon. And it can myelinate more than one axon. So here, in a way, it's more efficient because you have one oligodendrocyte providing for more than one myelin sheet, but because the cell body is so far away from the axon, if the axon is damaged, there is no regeneration. It cannot be regenerated, it cannot be fixed. So here are the oligodendrocytes. Here are the microglia. These are the white blood cells of the brain. So they're the ones that go through phagocytosis. They are sentinels. They're the sentinels of the cell. So they move around to look for debris and abnormalities. And also, in their resting stage, what they do is they prune off synapses that are not strong enough. So when we are sleeping, our microglia are highly active in pruning those synapses that were generated during the day, but that are not strong enough, they're not worth enough keeping. So they are pruned, they are removed by microglia. So they are very important cells, like all of the cells in the CNS are important, and their function is surveillance and to remove debris by phagocytosis. When I say processes, I mean the synapses. You mean, I don't know, I said processes that the microglia also have all these branches. So I don't know why was I saying processes? I've said it in so many different conditions. Okay, when I mean processing, it's in terms of neurons. Processes are axons and dendrites. Okay, just to make that clear when I'm talking about neurons. Okay, here we have the astrocytes. Astro means star. So they're star-like cells, and you see that they form a bridge between the neuron and the blood vessel. They can actually feed the neuron lactic acid, which is one of the components that can lead to production of ATP. They also get rid of excess potassium in the neighborhood. They get rid of excess neurotransmitters. So they also provide for a cleaning of the extracellular fluid. Okay, so we talked about oligodendrocytes, astrocytes, microglia. Now let's go to the ependymal cells. You see their shape is totally different from the others. So these ependymal cells, they form like sort of a barrier that that borders the canal, like in this, the spinal cord has a canal in the middle that is filled with fluid, and that fluid is known as the cerebrospinal fluid, and it's produced by the ependymal cells. Here you see a histological appearance. So the ependymal cells are dedicated to producing the cerebrospinal fluid. So the cerebrospinal fluid is another fluid that is present in the brain and that is used to provide for nutrients and also to get rid of metabolic products. So the CSF is another fluid that is present only in the central nervous system. I hear someone. Can you mute yourself, please? Okay, so 4 types of glial cells in the CNS. Astrocytes, oligodendrocytes, the myelinating cells, the microglia that are sort of the cleaners, but also have other important functions, and ependymal cells that produce the CSF. Now, in the peripheral nervous system, so I talked a lot about the Schwann cells, and you see here the Schwann cell, the nucleus. No, sorry, this is not the nucleus. This is an axon. And you see that the Schwann cell was able to go around and around and around. The outer layer is the layer that has the nucleus. Sometimes this can go around 100 times. So it forms a very, very thick insulator membrane along the axon. But, so this is one type of glial cells. The second type of glial cells are the satellite cells. Don't confuse these satellite cells with the satellite cells that are present in the skeletal muscle. That's different. Those are muscle cells. Here, the satellite cells, there are glial cells that are present in the peripheral nervous system, and they help nurture the cell bodies that are located within the ganglia. So the satellite cells are present within the ganglia. And here you see a rendition of a ganglion. So you see here these small circles are the cell bodies of the sensory neurons in a ganglia. And here you see the cell body with the satellite cells around it that is protecting it and providing nutrients. Our Schwann cells are many myelin sheets layered. Yes, one Schwann cell can go around the axon more than 100 times. What are Schwann cells again? Schwann cells are the cells in the peripheral nervous system that produce myelin. Okay, so here you see the comparison between the Schwann cell in the PNS and the oligodendrocyte in the CNS. You see how different the architecture is. So in the peripheral nervous system, you have one Schwann cell for each of 1 to 2 millimeter section of an axon. So this Schwann cell is totally dedicated to just this section of the axon in the peripheral nervous system. Here in the central nervous system, we see one oligodendrocyte with these projections that go in direction of more than one axon and more than one section of the axon. So very different architecture. And this different architecture contributes to the fact that the PNS axons, when they are damaged, they can be repaired. So there's repair here, but in the CNS, if an axon is severed, it cannot be repaired because it doesn't have the protection and all the signals and nutrition that is necessary because the cell body of the oligodendrocyte is so far away from the axon. Okay, so here, just to give you an idea, this is a Schwann cell. Now, the Schwann cells, here's an axon. You see this little piece here. That's an axon that's being enveloped by a Schwann cell. But the axon is not 100% covered by Schwann cells. There are some spaces between Schwann cells. These are known as the node of Ranvier. And these are very, very important to maintain the action potential propagation down the axon. I don't want to explain that now. I will explain it later. But just keep in mind, you see here, that the axon is not 100% covered by the Schwann cells. There are spaces between two neighboring Schwann cells. And those spaces, the axon is naked, let's say, is not protected by Schwann cell. And those are known as the node of Ranvier. Okay. So myelination starts at around 14 weeks of gestation and continues to about the age of 20. So that means that even in adulthood, myelination goes on. It's not completely finished until later in life when you're about 20 years old. And this, you can see very well why a baby cannot walk. It takes about a year or longer to be able to walk. Why? Because these long motor neurons are not myelinated. This doesn't work, unfortunately. I wanted this to work, but it doesn't matter. It just shows a wobbly baby. So the fact that babies cannot walk as soon as they are born is because they are lacking, their motor neurons are lacking myelination. Okay, so here you see a Schwann cell. At this stage, it's enveloping several axons. So that means that these axons are not going to be myelinated. They're unmyelinated. The Schwann cell here, its function is just to support the axons. But here, now you see an axon that is myelinated, and you will see that the Schwann cell approaches the axon and then starts enveloping and goes around and around and around, pushing all the organelles and the cytoplasm to the outer layer. The outer layer is known as the neurolemma, and this is only myelin, is made up of myelin sheath. So it's here you see sort of the movement. And I highly recommend that you look at this site because it shows you how myelination occurs. Now, in the CNS, here you see the oligodendrocyte with, in this case, you have 3 axons that are being myelinated. Sections of these 3 axons are being myelinated by the same oligodendrocyte. But the process of myelination is the same. The cell just goes around and around and envelops the axon. Okay, so there's many diseases, or a certain number of diseases that are associated with problems with myelination. First, multiple sclerosis. We already talked about this, that this is when it's an autoimmune disease where our own white blood cells destroy the myelin sheaths. And this, of course, has a major effect on the propagation of the action potential, because now this axon where the myelin sheath is damaged will not conduct the action potential at such speed and in such a proper manner. So that's why you have these problems in multiple sclerosis. And you see here plaques with an MRI. You can detect the plaques where their myelin did not is partially destroyed. Okay, this shows the same. Now, myelin can also be destroyed in the peripheral nervous system. So multiple sclerosis is an autoimmune disease that occurs in the CNS, but it can also occur in the peripheral nervous system. And here it is known as Guillain-Barré syndrome. And here this demyelination is reversible because it's at the peripheral nervous system. It involves Schwann cells, and the Schwann cells are very nurturing and they can replicate, and there's no scar being formed. So this condition here is reversible. Then you have other conditions that lead to the destruction of myelin. For heavy metals are highly toxic to myelin, like lead and mercury. That's why it's not good to have lead in paint. Lead is highly toxic. Then there are some bacteria that produce toxins that can also damage the Schwann cells. Remember, the myelin is produced by cells that are alive. If there are toxins that kill the cells, then that's not good. So diphtheria is caused by this bacteria, Corynebacterium diphtheria, that affects mostly the respiratory tract. There is vaccine, fortunately. Don't think that it's easy to make vaccines. It can take years. Okay, so here are the types of neuroglia, just to summarize. So in the CNS, we have astrocytes, microglia, ependymal cells, and oligodendrocytes. You should be now very well familiarized with these 4 types of microglia present in the CNS. In the peripheral nervous system, we have the Schwann cells and we have the satellite cells that protect the cell body. Okay, this is going to be recorded like your colleagues say, so we're almost done. Sophia. Okay, so this is the last concept. There's only 2 or 3 slides here. So, can the neurons be repaired? No, only the neurons from the peripheral nervous system. What happens is you see here the Schwann cells. So an important concept to know is that at 6 months of age, neurons, there's no more neurons, there's no more mitosis. So only some type of repair can occur, and that's in the peripheral nervous system. So let's see what happens. So in the peripheral nervous system, here is the axon is shown in blue, and it's severed. So you see that there's a lot of debris here, and the distal stump. So it's we're going to divide the axon into 2 different fragments. One is the proximal stump. That means that the proximal stump is contiguous with the cell body, and this is the distal stump that is away from the cell body. So why am I making this difference? The distal body has to be destroyed because it's not near the cell body, so it's impossible to recover. So it's going to be destroyed. But the proximal stump is going to be safe and it's going to be repaired. So first of all, what arrives at the site of injury? The macrophages, the white blood cells, to clear up the debris. Remember, here we are at the periphery. There's no microglia at the periphery. So at the periphery, what clears up the debris are the macrophages. And then you see the axon terminals start to be repaired, to grow. And the repair and the growth of this proximal stump of the axon depends on the viability of the Schwann cells. So the Schwann cells, if they're viable, they start dividing and they form sort of a tunnel that allows and guides the axon from the proximal stump to grow. So here are the 4 steps. You have the destruction, you have the clearing, you have the formation of the tunnel by the Schwann cells that are dividing, talking about the periphery. And then the axon is repaired. Now, the distal part of the axon is destroyed, is removed, and we call that the Wallerian degeneration. Wallerian degeneration refers to the destruction of the distal part, the part of the axon that is not in continuity with the cell body. But the proximal part of the axon continues and can regenerate and meet this, in this case, the muscle. So this happens at the periphery. So here, so there's also signals that come from the proximal stump, send signals to the cell body. And what happens is, at the cell body, there's a phenomenon known as chromatolysis. What is chromatolysis? Chromatolysis is the dispersion of the Nissl bodies. If you remember at the beginning of the lecture, I said that the Nissl bodies were groups of ribosomes that when there are signals from the proximal stump that there's injury, these Nissl bodies disperse and they are highly, highly active in producing new proteins. This is known as chromatolysis of the Nissl bodies. So they disperse and they become activated so that they make a lot of proteins that allow for the repair of the proximal stump. The distal stump is degenerating by Wallerian degeneration. Okay, so now here you see what happens in the central nervous system, disaster, because the oligodendrocytes are far away. So these are the oligodendrocytes. You see, we have the cell body, and they have these stumps that can myelinate just segments of the axons in the central nervous system. Here you see the astrocytes. What do the astrocytes do? They make a scar. So you have the clearing by the microglia and so on, but then the astrocytes make a scar tissue. And the presence of this scar tissue prevents the proximal stump of the axon to continue growing. So the astrocytes make the scar, and there's also a lot of inhibitors that prevent the growth of the proximal stump in the central nervous system. Okay. So here are the 2 processes, and this is my last slide. So Christopher Reeve, he was the first Superman in the movie, and he was highly active and in very good shape. He liked to horseback ride, and he fell in such a way that his column, his spinal cord, was severed between the C1 and C2 cervical vertebrae. So because the spinal cord this is the central nervous system, cannot be repaired, he, for the rest of his life, he was stuck in a chair. He had to breathe with an artificial machine and so on. He lost all different functions except for the head and the mind, because he started a foundation to do research for the repair of the axons in the central nervous system. Okay, you guys are so complainers. I finished here. So in the axons cannot be repaired in the central nervous system. Look here at the scenario. So here you have 3 axons and they were severed, right? Right here. And what do you see here? You see the oligodendrocyte, these blue, and these red are the astrocytes. So what happens? This is the distal stump and this is the proximal stump. Here would be the cell body. So at the distal stump, it is cleared by microglia and so on. But the astrocytes start making a scar. So there's a scar like a wall, connective tissue, fibrous tissue is made here so that this prevents the proximal stump from growing, to passing this scar. It cannot pass, so it cannot grow, and that's why it cannot be repaired. Why does the scar have to be developed? So it's a protective sort of mechanism to prevent the damage and so on from spreading to other areas. But there's so much research going on to try to overcome this big hurdle, which is the formation of the scar. It's not I usually focus more when I know it's Zoom over recording. Oh. If the scar tissue is to be removed, would regeneration of the CNS be possible? Well, removed, yes, but at the right time. So there's also a lot of inflammation going on, but if one could prevent the formation of the scar, that would be one step closer. No, it's because the astrocytes, Kevin, it's because the astrocytes make this scar, and the astrocytes are only present in the central nervous system. And one aspect is that the Schwann cells are in very close proximity. There's no astrocytes at the periphery, right? There's no astrocytes. Where is it here? See here, this is the PNS. There are no astrocytes forming a scar. You have the Schwann cells very close to the axon, and they provide a means for the repair. However, here, we have the astrocytes forming the scar, and we have, besides that, the oligodendrocytes that are far away. The cell body is not close to the axon, so it doesn't provide such a repair mechanism. Slide 46. This slide, you don't have it? Okay, I'll take a look at and see. If it's not there, I will put it in, okay? Thank you for telling me. Okay, not that one. Which one? It's a different one. But you said 46. This to me is 46. One of the repair slides. Okay, I'm going to go over them. This one. Wait, let me go back. This one. This one. Oh, this one. This is the one that's missing? Okay, number 44. Okay. Thanks for telling me. I will include it. It's a very nice slide, actually.
Excuse me, Professor.
Yes.
I just wanted to ask something. Like, I just got a question that got in my mind. So in Alzheimer's, it's mostly something that happens in the central nervous system.
Yes, absolutely.
So in the central nervous system, then does that mean that there's a lot of scarring happening, and that's why
Well.
Alzheimer's happens?
No. The scarring happens when the axons are severed or damaged, right?
Mm-hmm.
In Alzheimer's, the case is different. The scenario is different. What happens is that the neurons that produce acetylcholine, they degenerate. The neurons themselves, the whole neuron degenerates. Not because the axon is severed. We don't know why. Because of the conditions in that area might be toxic. There might be oxidative stress. There might be some kind of toxin that is present there. We don't know very well why. What is the initial? There can be many different factors that lead to the degeneration of the cholinergic neurons within that cause Alzheimer's. It has nothing to do with the severing of or damaging of the axon, like in this case, what I'm referring to. It's different. Is that
Yeah,
clear?
because I was like, just like looking like about the work you do with, like, how your research is with Alzheimer's, and I think it's like it's pretty cool, the work that you do, and it's really fascinating.
Yeah. It's this is a total different type of research where there's a lot of investment, because if you think about paraplegic and the people that have accidents and then become paraplegic or so on because of their spinal cord neurons being severed. So the effort is to try to reintroduce repairing within the central nervous system, in the axons. So one thing in Alzheimer's is to regenerate the neurons. Here is to repair the axon. So it's two
Mm.
different problems that are
Okay.
very hard. Okay, that's good
Yeah,
questions.
yeah, it's just very interesting to me.
Yeah, it's interesting to me too. I spend my life studying this. And I like to teach it also because I think it's very interesting. Okay, anything else? That's it? Okay, everybody. So today's Thursday, so I will see you Tuesday. We'll continue with this conversation. Nervous system to me is the most interesting. So have a good weekend. Bye-bye.